Hello everyone. Today we'll introduce BoltzGen, a protein design function in WeMol. BoltzGen applies an all-atom generative model to achieve unified structure prediction and generation, applicable to various systems including antibodies, proteins, peptides, cyclic peptides, small molecule receptors, and antimicrobial peptides. BoltzGen's experimental results demonstrate high consistency and versatility. This marks the transition of AI-driven protein design from specialized tools to universal platforms. Next, we'll introduce the operation of BoltzGen. BoltzGen in WeMol is divided into two modes: de novo antibody and custom. De novo antibody is primarily used for de novo design or modification of antibodies or nanobodies, requiring relatively fewer parameters and simpler application. Click the hint button next to each parameter to view specific requirements for input parameters. This model supports antibody and nanobody types. If you have an antigen structure, you can upload the antigen PD to antigen structure. If you only have the antigen sequence without structure, you can upload it to antigen sequence. In binding hotspot, you can set the residues on the antigen that participate in binding. Pay special attention to antibody sequence. This item uses an input format of sequence letters plus numbers. The sequence can use universally known framework template sequences, while the numbers represent the CDR region lengths of unknown sequences. It can also be left blank, and the module will use default antibody sequence templates for de novo design. Finally, you can set the number of design structures to output. Here's an example. Given a known antigen structure and hotspot residues, how to design an antibody structure de novo. Select antibody for type. Upload the antigen structure. Click the small eye icon to jump to the structure visualization editor WeView to view the structure and binding residue positions. Then select the antigen chain and set the binding sites. This completes the input parameter setup for de novo antibody design, and you can directly run the task. The custom mode function in BoltzGen is very powerful and flexible. Users can customize many parameters. It can be used for protein, peptide, small molecule binding protein, and nanobody design. Custom mode supports three types of design rules: definition based on existing structures, definition based on sequences, and definition based on small molecule files. The three methods can be freely combined and used together. Set input parameters and files according to input prompts. Upload existing protein structures in structure. Can be used to extract target proteins or extract partial structures for design modification. Chains can be extracted as target proteins or for modification in chain. Include is used to further specify the residue range to extract based on the specified chain. Exclude is the opposite of include, specifying which residues not to extract based on the specified chain. Design position specifies residues that need to be designed based on the extracted structure. Note that residue numbers to be redesigned should exist in the extracted structure. Design SS can specify secondary structure types for residues to be designed. Binding hotspot is used to specify residues involved in binding. Note that these residues cannot be residues specified for design modification in Design SS. Non-binding is the opposite, specifying which residues do not participate in binding. Design insertions is used to specify insertion mutation design. Sequence can specify the sequence to be designed. It can include target protein sequences, peptides, or proteins that need to be designed and generated. Sequence binding is used to specify residues in the sequence that participate in binding. Sequence non-binding is used to specify residues in the sequence that do not participate in binding. Sequence SS can specify secondary structure types of residues in the sequence. Sequence cycle can specify sequence numbers that need to be cyclized. Ligand can specify small molecule information involved in binding. Covalent bond can specify covalent bond information in the sequence. Number of designs allows you to set the number of design structures to output. After introducing the input parameters, let's move to the practical demonstration. Case 1. Given a target protein structure and binding site, design a protein to bind with it from scratch. Select protein in protocol. Upload the target protein structure. Select the chain of the target protein. Input the binding site on the target protein. In sequence, input the approximate length of the protein to be designed. The input parameter setup for this case is complete. You can submit the task for execution. Case 2, given a protein complex structure, modify the ligand protein. Still select protein in protocol. Upload the protein complex structure. Select chains, whereas the target protein and B is the ligand protein to be modified. Extract the residue range for modification from chain B. Further specify the residues that need to be redesigned. Assign secondary structures to the redesigned residues. Set the known ligand binding sites. The input parameter setup for this case is now complete and ready for submission. Case 3, given a target protein sequence, design a binding cyclic peptide. Select peptide in protocol. In sequence, input the target protein sequence. Then input the designed cyclic peptide sequence on a new line. The example cyclic peptide is based on an existing sequence with 4 to 5 residue insertion mutations after the first residue. Input the known binding sites on the target protein. Fill in the cyclization information for the second input sequence. Fill in the disulfide bond information for the target protein sequence. The input parameter setup for this case is complete and ready for submission. BoltzGen tasks typically take over an hour to run. After task completion, the designed complex sequences are output, including batch format complex sequences and the final design chain sequences. A compressed file containing all design structures is output. A scoring file for all design structures is provided. The final rank in the scoring table is the final ranking based on comprehensive scoring of various indicators. The meaning of other subscores can be referenced in the result description in the introduction documentation. By clicking the book icon in the report interface, you can view the detailed documentation of the module and understand the output result parameters. The top 5 example structures are provided after the table. We can use WeView to open and examine the specific structural details. That concludes this video. Thank you for your watching. Welcome to try WeMol.